From Microbe TV, this is Office Hours for Wednesday, September 6, 2023. I'm Vincent Racaniello, and welcome to the viral corner of the internet. Welcome, everybody. It's been a couple of weeks. We missed 2 Wednesday night sessions because I was traveling. Last week I was in Milan. The week before, I think I was in Vermont. All for podcasts, of course. So it's good to be back. I'll be back, I think, for a couple of weeks now. I don't think we're missing one this week, next week, the following week, the following week. Yeah, you're not going to be missing one until, I think, November-ish. Still have a bunch of travel on slate for the rest of the year. I'll let you know a little bit about that. But I want to first thank our moderators for being here tonight. Really appreciate your work. So we have Les, we have Barb Mack, we have Tom, we have Barb Mack UK. Frank is here tonight. Good to see you, Frank. Hope all is well. And Vanity Nutrition. Thank you so much for being here and keeping the conversation civilized, as we say. Let's see, who do we have here tonight? Where are we from? I haven't done that in a while. I know where all of our moderators are from. Nicola is from Italy, where I just was last week in the wonderful city of Milano, where I spent all of 3 nights and didn't see a darn thing, but I got to eat good food. I think Barb Mack, of course, is from the UK. Let us see. Wandering World may be from Edinburgh, right? I know Lisa's from Ohio. Will from China, Changsha, I believe, and I will be visiting Changsha somewhere around the 11th to the 13th-ish of November. I have a invitation from a virologist there to give a talk and to visit. So I'll be in Hong Kong from the 8th to the 10th, roughly, maybe the 7th through the 10th. Changsha for a couple of days, then I'm going to go to Hangzhou for the rest of the time. I'm trying to figure out how to get back to the U.S., whether I should go to Shanghai or back to Hong Kong, but I'll sort that out. Anyway, welcome. Hello, Jessica from the University of Toledo. Good to see you again. I think a lot of people miss this, which is great. I love it. You know, I'm there traveling, doing my thing, and I'm thinking, man, probably nobody's even thinking of it. Who else we got? Noir is New Mexico, I believe. Where is Amy? Amy will no longer be here, I'm sorry to say. As a government employee, she can't do it. Boulder, Colorado. South Carolina. Cool. Very cool. Ontario, Canada. Coxsackie, New York. Oh, I'm sorry. Hope you're doing well. Your microscope is right here on the floor next to me. I have your address. I will pack it up one of these days. I have to stop traveling. Tomorrow I'm going to Salt Lake City. I'll talk about that in a minute. Like, will the start time remain the same? Yeah, I'm going to leave it at 8 p.m. You know, I was thinking of making it 7, but I decided not to. Urbana, Illinois. Miami. Doreen, somewhere in Illinois, I believe. Ooh, Ukraine. Dean, how's it going out there? Hope you're safe. Kang is from Chicago. And Kang, I'd love to have dinner with you. I'll let you know when I'm going to be there. I haven't settled my dates yet. John is from Minnesota. Wanda is from D.C., the District of Columbia, Wayne County, Ohio, West Virginia, Las Vegas, Rhode Island. I love doing this, folks. I hope you don't mind that I go through and see where everyone is from. East End of Long Island. Had a visitor at the incubator tonight from Long Island, and that would be Rona, who is often here, but she's not here tonight. Winona, Minnesota, Hampton, Virginia. Belo Horizonte, Brazil. Cool. Great. I love Brazil. I love Brazil. Some good questions I'm seeing here. We'll get to them. Hello, the two San Francisco PharmDs. Thank you, Kip and Laura from San Francisco. Really appreciate your support of Microbe TV. Queens, New York. Yep, 1964 World's Fair. I went to see it with my family. I remember that very well. Jan is somewhere, I think, in Arizona. Portland, Oregon. Yeah, Tucson is back to 40C. New York City today. I don't know if any of you were in the city. The sky was absolutely blue and clear, and the sun was beating down. It was so cool. Not cool. It was really hot, but I love that hot weather. And at 10 a.m. when I got to the incubator, the sun was beating down. It was just amazing. Just amazing. No more Amy. I'm sorry to tell everyone that she's no longer allowed to participate. And I really, it's unfortunate because I think she was a great addition. I really enjoyed interacting with her. Amy has truly become the plaque assay princess. So let me show you the plaque assay princess. There she is. It's a present of Barb Mack. A nice drawing. Let me make it a little smaller so that we can see it in its full glory. So we have the slippers and we have the lab coat, the name tag, the plaque assay with viruses, the princess crown, and the cashmere sweater, of course. So thank you, Amy, for all of your contributions here. This is very funny. Amy took the king's shilling. Good on her. Good on her. I don't know what it means to take the king's shilling. It's really funny. I like that. St. Cloud, Florida. If Amy's listening, everyone's very sad that you're not going to be here, and they're thanking you. Okay, so this is a flag. I don't know where it's from. Well, I don't know. It's either Spain or Portugal, I'm thinking, but I don't know. Yeah, who's in for seeing Dr. V in Chicago? I'll tell you right now, in the days of the ASTMH, you can always go check it, but ASTMH is from October 18th through October 22nd, okay? You can just search ASTMH and you'll find that. So there's quite a few days. It's like Wednesday through Sunday. So you can check it out. If you want to get together, yeah, that would be great. Okay. That's it for to take the king's shilling means to enlist in the army, the shilling being a reference to the earnest payment made to each new recruit. Mexico. Sorry, Israel. That's probably offensive that we got your country wrong and that we're so ignorant that we don't know. At least I am. We don't know country flags. Welcome. Okay. Let's go back to the top. I wanted to tell you a few things before I dive into your questions. All right. Let's close Israel here. So, upcoming travels. Well, you can look on the travel thing, but tomorrow I'm going to Salt Lake City. This is quite interesting. There's a retrovirus conference over at Snowbird, and they're putting on a play, a science fiction play based on retroviruses. So I'm going to interview the playwright, the director, the producer, and one of the actors. And that should be, we're going to talk about the play, and then we're going to see the play put on. It's just going to be a reading, but I'm going to film it and incorporate clips into the TWIV episode, so that should be fun. Saturday, I'm going to go see Nels. And so I have to post this tomorrow. We're going to do a live stream together Saturday from Salt Lake City. So TWIVO, Saturday live stream. I have to remember to put up the thumbnail. I don't know what time he wants to do it yet, but the plan is that we're going to both sit in his office in front of the whiteboard with all the squiggles on it and do a live stream. That should be fun. And then he's going to take me on a little hike because he said I need to get some fresh air. Okay, so we're going to do that. So that's one thing. Second thing, a couple of TWIVs ago, we had Jolene Ramsey, who, by the way, I think is fabulous. When I heard her talk at ASM, such a presence, knows her stuff. So I said, you got to come on as much as you can and do a phage paper. So she's going to come on like twice a semester. Anyway, I thanked her for doing the timestamps, and I said that Les gets the timestamps from Jolene. But then I realized after I said that that doesn't work because the video and the audio are different. So I edit the audio, and I often cut things out that I don't happen to like at the moment of editing. There are no rules or anything, but that's what I do. And so the video is done by David Renada separately, who doesn't cut anything out typically unless he sees something and he asks me. So the timestamps that Jolene makes for the audio would not work for the video. So just so you know, I misspoke, and I'll announce this on TWIV again. Okay, next thing. I want to show you some photos of my travels. Okay, so where have I been? Let's see. Can I see a preview of these things? Okay, so this is Baltimore, Maryland. You can see it's windy because my hair's messed up. And here, for the first time, you can see that I actually have some colorless hairs, okay? Not gray. They're just colorless, folks, because here in the live stream tonight, you can't see them, right? And I don't color them. They're just in there buried, but when it blows, yeah. So anyway, these are two, I believe, PhD students. I was waiting for a taxi, and they ran out and wanted a photo and an autograph, and they're at the CEIR, CEIR, Center for Excellence in Influenza Research and Response. It's a center funded by a large NIH grant that gets people from different institutions together to work on influenza. And they meet once a year. And so I got invited to do a pod, and I did one with Ron Fouché and Louise Moncla and Raphael, I can't remember his name, but we all talked about avian influenza, and that'll go up in a couple of weeks. So if you're worried about avian influenza, you should listen to them. And then, okay, so that's that. Now, what's cool about this photo, folks? Obviously, this young lady here is smiling, right? Great smile. But this lady with the mask, you can still tell she's smiling, right? Because her eyes are smiling. I just love it that you can tell that someone is smiling even with a mask on. You don't have to see their face, okay? I just think that's great. Okay, so that's Baltimore. Now, here is the convention center in Milano. This is at dusk, right? I'm walking around. I believe I just had dinner, and I'm walking it off. Isn't that an interesting place? It's in a part of Milano that's, I don't know, there's a lot of residential stuff, and they built this convention center, and there are a bunch of hotels. It's a little uninteresting place, but I thought the convention center is cool. Okay, so there's that. What else do we have here? Okay, so this is I know who this is because she started following me on Instagram, and so I can find her. Where is it? Oh, I know where it is. It's in the other place, yeah. Her name is Lucy Vranova. She is from Slovakia, I want to say. Anyway, a number of people at the meeting came up to me. This was the European Society for Clinical Virology. And clinical virology is viruses having to do with people, diagnostics, therapies, right? Not a lot of basic fundamental stuff, a little bit of immunology. So you get a lot of laboratorians, people who run clinical labs, diagnostic labs, people who study vaccines and so forth. So she was there. That's cool. And I didn't think anybody would know about TWIV, but of course there are many people who knew about it. That's pretty cool. All right, that we saw that already. And then, all right, this is from Baltimore. I'm skipping around, sorry. But Karen, my assistant, had this made. It's a pull-up thing, right? It's called a pull-up banner. And we bring this to shows and put it next to the table. I think it's cool, right? We got our experts here. And, of course, we don't have Jolene. Unfortunately, it was made just before she got on. But I'm going to have to print out a white one and put it on here because we can't. These are $250, and I don't want to make another one. Okay, so that's that. And I think that there was one more picture. Where is it? Here we go. So this has a story behind it. So these are 3 virologists from Belgium. And this is the convention center where the meeting was. Big industrial type space. Very interesting. A lot of room. Pretty good room for the talks. They had separate room for TWIV. And off in Belgium, Elke, this is Elke Volant here. So Elke, a year ago in Manchester, I was leaving the convention center, and she ran after me. She said, "Where are you going? I need a picture with you." I said, "Oh, I'm sorry. I have to go to bed because I have to get up at 3 a.m. for a flight." So we took a selfie, and then I took a picture of her badge. I said, "I want to shout you out on TWIV." She said, "Oh, you'll forget about me tomorrow." So I made sure I shouted her out. And then she came up to me at this meeting. I said, "Your name is really familiar." She said, "Yes." But on top of that, what this group does is when planes land, so they did this study on airplanes coming from China. The plane lands, what do you have to do? One of the things you do, you have to refuel it, you have to clean the insides, blah, blah, blah. You have to remove the toilet waste, right? So they put a hose onto the outlet on the side of the plane, and they pull out all the waste. Well, she and her team collected it from a number of flights coming from China. So they go from China directly to Belgium, and they say, the people on the plane, what do they have in them? So, oh my gosh, what do they have in them? I wrote down what they have in. Anyway, I want to get her and maybe some of her colleagues on TWIV, because as soon as this is published, because it's a fascinating thing. You can monitor what these people have. And so, here, let me find it. Where is it? Of course, they had SARS-CoV-2. No question about that. Plenty of SARS-CoV-2. Here we go. Where is it? I know they found enteroviruses, but they did not find poliovirus. Where is it? Why can't I find it? I guess I should have marked it, right? Neonates, Hagemans, harms. It'll be worth your wait. Hang on. So that's Elke. What's what she did? And now she's going to extend it to planes coming from countries all over the world, and it can give you a clue about what's coming in. That could be useful, right? I just had it before. This is really unfortunate. Massacre. No, it was before massacre, or maybe it was after. I can't find it. So I think I remember. Human papillomaviruses. Remember, this is coming from urine and poop, right? Papillomaviruses, adenoviruses, SARS-CoV-2, influenza virus, I believe, and others. So it will be interesting to see what else is coming from other countries and whether you can use this to give you an idea of what's coming in to your country, because then, these people enter the country, and if there's a little outbreak, you can maybe get a handle on tracking it. Oh my gosh. I'm really mad. I just saw this in this stinking little field notes, which is supposed to make life easier. And, I would have written Woolance at the top of the thing, but it's not there. Okay, anyway, that's that. So enough of that. Those are the pictures. How do I turn this off? There we go. Okay, next thing. I mentioned travel already. And I gave you... I want to tell you a few more things about ESCV. There's some cool stuff going on. Okay. And then, of course, I'm going to come across Elka's thing. Well, maybe it's in between the podcast stuff. Here it is. I didn't look far back enough. So flights from China. They offloaded the toilet waste. 60% of the samples were positive with high CT values, which means not a lot of nucleic acid there. And this is interesting. So they found 60% of the sample positive for SARS-CoV-2, even though you had to test negative to leave China. So I don't know what kind of test they're doing there, but obviously there's a discordance, and maybe there's not much nucleic acid in the stool anyway, or in the waste. They were all Omicron. And they found no variant of concern that had not been already in Belgium. So that's an interesting thing. Found polyomaviruses. So by the way, any Jimmy Buffett fans, right? He died of Merkel cell carcinoma. It's a very rare cancer of the skin. It's an unusual cell type in the skin. And that it can be made cancerous by Merkel cell polyomavirus or UV light. So they found polyomaviruses, human papillomavirus, noroviruses, enteroviruses, adenovirus. They found bacteria, all poliovirus negative. Yeah, that's really interesting. I'm surprised that there was no poliovirus, because if they're using IPV, then OPVs should be circulating. And if they're using OPV, you should find OPV. So I don't know why, what's going on there. There was a talk on, an outbreak of H5N1 avian influenza virus in Eurasian otters. And the one they showed a map, and Australia has no H5 infections, and we know that already. I've known that for a while, but it was just interesting, really, really interesting. Okay, then there was this very sad talk of a person who got a heart transplant from a pig. So he needed a heart transplant, and he signed up for an experimental one. Listen to this. So the pig has in its genome something like how many pervs? You folks know what a perv is? Do you know what line what podcast that line is from? Do you know what a perv is? Anyway, porcine endogenous retroviruses. They're retroviral sequences integrated into the porcine genome. So they knocked them all out, 60-some. Then they knocked in a bunch of genes. They did this in stem cells, and then they did stem cell nuclear transfer and derived pigs, put them in the pigs, derived pigs, and made sure they were raised under sterile conditions. They were checked periodically for any pathogens. They bred in a pathogen-free facility. So they give this man the heart of this pig. And on day 20, he tests positive for porcine cytomegalovirus. And the pig had been screened by nasal swab for porcine cytomegalovirus. And it was negative, but obviously the virus was elsewhere. possibly in the blood. Anyway, it's not what killed him. He had gotten intravenous immunoglobulin, I guess, to protect him against infections because he was being immunosuppressed, right, to prevent rejection. And the intravenous immunoglobulin caused antibody-mediated rejection of the tissue of the heart. So the heart was destroyed, and I mean, he was just being maintained on life support, right, for weeks. And he said, that's it, pull the plug. I don't want to do this anymore. And they showed a picture of him at the end of the talk. You know, it was really, it's very moving because he had a picture of himself smiling, and he was 57 years old. And he said, I hope what you've learned contributes to progress and can help other people, right? I just think that's great. So just a few more things I want to tell you about, then I'll start getting into your questions. But I think, you know, you might as well benefit from my experience. There was another interesting talk. So you guys know about hepatitis E virus. Hepatitis E virus. This is a virus that is, well, it's in, you know, it's in the series of hepatitis E virus. It's an RNA virus. There are 4 serotypes. 1 and 2 are endemic, and they're waterborne. You can get them. And 3 and 4 are zoonotic and sporadic infections. So wildlife have these viruses, and certain meats that you eat can be contaminated with it, and they will cause hepatitis. Anyway, in this case, it was a 59-year-old heart transplant recipient who had a history of close contact with his neighbor's rabbits. He had this. He was diagnosed as Hep E positive, and he was infected from 2012 to 2021. Got his heart transplant in 2012, and shortly thereafter developed this infection, presumably because he was immunosuppressed. Treated with ribavirin, but was not successful. I don't know what happened to him, if I remember this, or not. So hepatitis E virus. And there was another talk by someone else who said that they did a study in the Netherlands, and they went into the supermarkets, and they found hepatitis E viruses in the meat, and those viruses matched what blood donors had given. So in other words, people bought the meat from the supermarket, they ate it, they gave blood, and they gave the virus in it with the blood, and the virus in the blood matched the viruses of the meat that they ate. Isn't that funny? Anyway, there's another talk about another transplant patient who got a transplant in 2006, ended up getting infected with hepatitis E virus, died in 2020. Virus went into his CNS. Final thing. The final thing, this is very interesting. Peter Openshaw, an immunologist at St. Mary's Hospital in London. I sat next to him for dinner. Very interesting fellow. It turns out that people who get frequent respiratory infections throughout their lives have earlier deaths than people who do not. Okay? And the risk, you have a twofold increased risk of death if you have had more respiratory infections in your life. The risk is almost as bad as the increase in death caused by smoking. Okay? So if you have a lot of respiratory infections, and this was published in March of 2023, and I have to look it up. I haven't found the paper yet. If you get respiratory syncytial virus, it has lifelong effects. It can cause asthma for your life. So that's a long effect as well. I'm just saying this because he was saying, there are a lot of viruses that do long things. And in fact, pavlizumab, the monoclonal that was used to treat respiratory syncytial virus infection in infants, gives a 61% reduction in wheezing illness, but it does not reduce atopic asthma. And so he wondered whether the better monoclonals and the vaccines are going to impact that in any way. Long-term survival of adults, they survive shorter after respiratory syncytial virus than with influenza virus. Okay, so lots of long-term effects with respiratory virus infections, not just with SARS-CoV-2. Now, he did talk a lot about SARS-CoV-2. He talked a lot about MECFS as well, and cited a study. Reduced COVID outcomes, long COVID outcomes after mild COVID. And what was the other one here? I had seen it before. All right, that's it. But so look up Peter Oppenshaw, very interesting fellow, and very interesting dinner conversation with him as well. Let's do some questions here, folks. I hope that was useful to hear. So you should get RSV vaccine. Had a great discussion with Paul Offit today about vaccine side effects and how if you do a study, a safety study with 50,000 people or a phase 3, you're not going to see rare effects that are 1 in 100,000 or 1 in a million, right? There's just no way you're going to do that. You can only see that afterwards, after licensure. So I said, so can people make their own decisions, though, if they don't want to take that risk? He said, well, there's always some risk. If you don't get vaccinated, there's a risk. And if you do, there may be a smaller risk. So you have to be smart about it. I think that's the way to look at it. Anyway, that's the story. So there's a question here from... Okay. So Polio Pete, hello Polio Pete, said, I think office hours would outcompete tomorrow's NFL game. I said, no. Even though, we're 2 hours, people probably say, it's too long. But TWIV, of course, is 2 hours, and people say it's too long, but they watch a football game. James says, those who pick their nose are 3.8 times more likely to catch COVID than non-nose pickers. Yeah, that's absolutely right. It's absolutely right that if you pick your nose, you will have a higher incidence of COVID and other things. Well, you don't catch COVID, you catch SARS-CoV-2 infection, right? Let's just be pedantic. So what is picking your nose? It doesn't have to be, sticking your finger all the way up. It can just be touching the anterior nares, which many of us do as kind of a reflex sort of thing. And then you get contamination with mucus, right? So try not to do that. Or if you do it, wash your hands before you touch something else. I'm glad everyone missed this program. There are lots of comments here saying that people have missed it. Thank you very much. John has finished Journey from all the TWIVs from the beginning that are available. They should be from 1 to 1,041, right? They should all be available. Not on YouTube, because we only started doing video recently. But the audio is certainly available from 1. Now, it may not be available on your favorite podcast program for technical reasons that I don't need to get into, but it's certainly available on the webpage, microbe.tv. You can go all the way back to number 1. You should go to 1. The early ones are fun, too. Anyway, thanks for listening. Appreciate it. To all of them. If they keep updating the COVID vaccines every year, will it always be under an EUA? I don't see why it has to be. Is that what they're doing now with the? I don't know the latest. The bivalent booster, which is what's being used now. Of course, we're going to get a new booster, a non-bivalent, soon, with one of the more recent variants. Not the most recent, but one of the more recent. Is that an EUA? All right, it's because with influenza they don't do that. They have licensed influenza vaccines, and they update them every year with minimal testing, and that's not under an EUA. So I don't know why it would have to be for COVID vaccines, right? Unless it completely changes the virus. But even then, you could argue that the first booster was very different and should have been treated differently, but it's fine. So I don't see why it would need to be under an EUA. It should be a modified version. If you're just updating to a variant, you just do minimal testing, antigenicity, immunogenicity, and that's it. Shouldn't be a problem. How does an electron microscopist distinguish a novel virus from an artifact? Well, so what happens most of the time is that you look at the picture of the virus under the microscope, and you see if it matches something. Like coronaviruses have the unique corona-like appearance, right? So you say, okay, this is a coronavirus. Influenza virus, noroviruses, polioviruses, adenoviruses, herpes viruses, they have distinguishable morphology. Now, if what would an artifact be? Something that doesn't look like anything that you know. It would be hard because, all viruses fall into certain morphological groups. And so, pox viruses are very distinctive. The giant viruses, remember Mimi virus, not only was it icosahedral, but it was fuzzy, right? It had the fuzzy hairs sticking out of it. Everybody knows what that looks like, right? Okay, let's take a look. We can get a nice, this is beautiful. Very nice. Let me do a screen share here. Where is my screen share? Come on, screen share. Here it is. All right, so you see the nice icosahedral capsid with the fuzzy business around it. Here's another one. That's pretty cool. Here's more of a stylized one. Okay, so that's Mimi virus. And when they first found it, they said, well, it's an icosahedral virus, but we never saw one with those hairs. So then you have to characterize it, right? But what if it just looks like a sphere? Well, there are a lot of membrane vesicles in cells, right? Like exosomes and smaller vesicles within the cell that are involved in transport. So it could very well be that this is a virus, or it could be a cellular vesicle. And so sometimes you have to purify them and see if there's nucleic acid in them, see if they're infectious. It's hard because you don't know what kind of cell. So it's tough. If it doesn't fit into the known categories, you have to do some work. I would say you try and purify them and see if there's something inside of them. And if there's nothing that looks like a genome, then it's something derived from the cell, most likely. Any news with COVID? Well, as you know, we're in a phase of higher numbers of cases, probably because people are traveling now, bringing kids back to school, taking vacations. The airports are pretty full, at least when I was traveling. People are getting together and they exchange viruses. All right, so that explains the increased cases. And as I said, who did I say this to? I asked one of my guests on some podcast, what's the seasonality? And they say, well, we don't know what the seasonality is. It's not obeying any rules. When we don't have a lot of population immunity, basically, we don't get good seasonality patterns. So, for example, the 2009 influenza pandemic, it started in the summer, right? It started in August or something like that, just not the right season. So when you don't have a lot of population immunity, that whatever the effects are that control seasonality were out the window. And so that's the case with COVID until we have a lot of population immunity. So I don't know what it's waiting for, frankly. But anyway, what's new? There's some new variants around. I saw some data at the meeting in Milan that suggested that bivalent vaccines would do well against the variant in terms of neutralization. There are all kinds of premature proclamations about virulence and transmission. I don't even listen to them because it's not possible to make any reliable conclusions about those properties within a week. The only thing that's important is whether antibodies neutralize them. If they don't, you'll still be protected from severe disease and death by T cells and K cells, other mechanisms, unless you are sick and you have other issues. Then the infection will go out of hand, and that's where you need to take Paxlovid or remdesivir or molnupiravir. All right, so that's what's happening, and I presume there's going to be another burst of activity as we get into the winter months, right? So there's going to be a booster, which a very small fraction of the population will take. It will boost those antibodies for a while, which will give them some protection in the upper respiratory tract. And those antibodies will decline by the spring. And but most people won't take them, and they'll either be fine, or if they're over 65 and have comorbidities, they'll take Paxlovid. And I think that's the way it should be. I don't know about making a variant-specific booster every year. I'd like to see data that say it helps, right? So this year we're going to have a variant-specific booster. Let's collect data and see if it makes a difference. The only problem is we can't do a controlled study because we're only going to offer that to people. We're not going to offer people a bivalent booster. Right now, that's all you can get. So it's not great data. Does the HPV vaccine provide sterilizing immunity? I have not seen. Now, if you ask a virologist, they'll say yes, right? And I would have said yes 2 years ago, 3 years ago, for sure. Why? Because it prevents 100% of cervical cancer. And so the assumption is it must be sterilizing. But no one has ever checked, because to do that, you need to swab the cervix and look for a virus. Hard experiment to do by infectivity methods, which are very burdensome for human papillomaviruses. So I would say there's no evidence that it's sterilizing. It's good at preventing disease. That's fine, but that's not the same as infection. As you know, not the same. Okay. Is it plausible to think that the new mRNA COVID vaccine will be just as safe as the original? It's a good question. It sounds like a lawyer. Is it plausible to think? So the variant-specific vaccine, which is going to be released, new COVID booster. So what is the name of that guy? As early as Friday, huh? NBC could, FDA could green light as early as Friday. But I want to know what XBB 1.1.5. So that's going to be it. And, that strain is no longer dominant. And that's the problem you have when you're going to pick this in the summertime. By the fall, it's not dominant. Earlier than the summer, because you need time to make it. But apparently the antibodies induced by it can neutralize the currently circulating variants. That's fine. Okay, so will it be just as safe? I don't see why not. It's basically a spike protein. However, it is a different spike protein, right? It has a different amino acid sequence. Not 100% difference, but maybe mainly differences in the receptor binding domain, which impact antigenicity, the induction of the antibodies that you need to protect you against infection. So, I would speculate. Yes, I would. I would say it's plausible to think that it would be just as safe. But even if you did a trial with 40,000 people, it might not be enough to pick up very rare events. So you wait until you give it to a lot of people and see what happens. But, and people, if you're worried, don't get it. But I'd say you have to balance your risks, right? So if you're older and have comorbidities, you should consider getting it because it'll give you a boost for a couple of months. But other people don't need to do that. And if you're concerned about the risk, you have to balance those two. So if you're 90 years old with diabetes or heart disease or kidney disease, then you should consider taking it, right? Because your risks outweigh the risks of any problems with that vaccine. So that's my answer. So the new variant, so their new names, they also have other names, right? There's a guy who decided they need more human names. It's BA.2.86. And, these are viruses that have arisen after the virus is circulating and changes in the spike protein accumulate. And this one, there's also another one called Eris, right? EG.5. Pirola has over 30 amino acid changes, which, so Omicron XBB 1.5 to EG5, one or two mutations, but now we have 30 from Omicron. WHO says there's low global risk, probably because it's still neutralized by sera. First came up in Denmark, been detected elsewhere now. And, I don't think it's particularly concerning, but I don't think any of the variants are particularly concerning. But it's not very different in terms of antigenicity. And as I said earlier, I wouldn't worry about virulence because that's really going to depend on the population that's studied. But looks like it can be neutralized by the XBB 1.6 whatever. So that's what I think. I wouldn't worry about it. If you're in the risk group, get the booster. I think we skipped Carol, Las Vegas, our favorite nephrologist from Las Vegas and also Rhode Island, right? Okay, let's see. Did I understand that none of our vaccines prevent infection but prevent disease? That's correct. So, and it also depends where, when, how long after immunization are we talking about? Remember, within a few months after getting a vaccine, you have high antibody levels, let's say for a respiratory virus in your respiratory mucosa. Those will impede infection. I'm not sure they will ever be sterilizing, but they may reduce infection compared to when you have lower antibodies in the mucosa, right? But they don't block infection. They will prevent disease from happening. And even when those antibody levels in the mucosa decline, when you get infected, the virus will begin to reproduce, but then you'll have a memory antibody response, right, that produces antibodies that will neutralize the virus, so it will prevent it from spreading and causing extensive disease. And it's only if there's an antigenic mismatch between the memory-induced antibodies and the virus do you have potential issues where the virus is going to reproduce more, and in people who have health issues, they can get very sick. So yes, none of the vaccines prevent infection. And you can go, as I said, you can ask any virologist, and they'll probably say, yeah, the HPV one does. They may say the measles one does. But frankly, I would reserve judgment until I saw the study, because nobody's done the right study. Nobody has looked for infectivity at different time points after immunization. It's a hard experiment to do, right? You'd have to immunize a cohort, and then you have a control group. And then you basically have to swab them at least once a week to see who gets infected, then compare the rate of infection in the vaccine versus the unvaccinated group. And that has not been done with HPV. That has not been done with measles virus. So they prevent disease. Polio vaccine was developed in the 50s. What was the readout for the clinical trial? Reduction of poliomyelitis. For every vaccine, that's what they're looking at. That's the primary endpoint. For the COVID vaccines, it was reduction of COVID, not infection, not PCR positivity. So that's correct, Patricia. They prevent disease, not infection. Okay. Alina's day 4 with Paxlovid. All right, good for you. Hope it's good. I had Paxlovid on day 2. Symptoms went away the next day. It's good. Aside from COVID, are there other known viruses that affect taste and smell? I think it's funny that you say known, right? Because we wouldn't know it. I'm feeling kind of like, what's the Secretary of Defense? What was his name? The known knowns and the unknown knowns and all that. Anyway, influenza viruses can affect taste and smell. Any other ones? You know, when I was in college, my first year in college, I had a respiratory tract infection and I couldn't taste anything. The food in the dorm was horrible. And then when I got my sense of taste back, it was still horrible. Rhinoviruses can do it. Other common cold coronas, para-influenza viruses. Post-viral anosmia. So I'm looking at a review article in the journal Lung, and it's entitled, it's published in 2021. Post-viral anosmia did not begin with COVID-19. You know, I love articles like this because people often think that everything begins with COVID-19. Here we go. I'm going to show you this paper here. All right. Here we go. Rhinovirus, coronavirus, parainfluenza virus, and Epstein-Barr virus. So there you go. It's been around for a while. Although, you know, in a pandemic, a lot of people infected, so you can imagine that it gets more attention, right? Totally reasonable. What else do we have here? Any questions? Taking the Kings, whatever. Here we go. Carol. I thought I understood antigen sin, but not sure. The protection against severe disease with the third shot, is that explained by antigenic sin? No. So antigenic sin means you have an exposure, your first influenza virus exposure. All right. And then some years later, you have another exposure to a related influenza virus. And most of your immune response is against the first one that you were exposed to. There's enough cross-reactivity between the two viruses that to the immune system, the new virus looks very much like the old one. So you're stimulating memory cells from the original infection to become activated. That's original antigenic sin. And so with COVID vaccines, many of us got the ancestral vaccine, and then if you got the bivalent booster, most of your immune response was against the original spike. Now, the third COVID shot, what that did was to diversify the antibody response. It stimulated somatic hypermutation and affinity maturation, which means that you now select for higher affinity antibodies that have a higher affinity for the spike and are presumably better at neutralizing infectivity. And that didn't happen with the first two because they're only three weeks apart. It's not enough time to get this affinity maturation. So the third shot is what did it. And then we saw that the antibodies produced after the third shot were really good at neutralizing all of the variants that we were seeing up until that time. And then, of course, came Omicron, which evaded even those and changed the story. But that is affinity maturation, not original antigenic sin. Okay? Yep. Why isn't Paxlovid given to people in their 20s, 30s, etc.? It's if you read the FDA package insert, it's for people who are at risk for developing severe disease. And so they're not typically, although they can at that age, but most cannot. And I was just reading it today. The insert says people at risk for developing severe disease. Do I still have this in my memory? Let's see. Probably not. And it's simply a numbers game, right? They don't need to get it. Of course, you can get it off-label if you want to. I know many people in their 20s and 30s who have gotten it. And that reminds me, those of you from Australia, I don't know why Martha reminded me, but I'm going to be going twice to Australia in 2024. Twice. I think once in the summer and once in June-ish and once in September. So one of them is going to be Cairns or however you say it, and then the other will be somewhere else. I do want to visit Sydney and Melbourne and do a couple of pods and talks and so forth. So look for information on that. So those of you who are disappointed that I didn't go this year, twice next year. So I should be able to see some people. That would be cool, right? Hello, Mark. Good to see you. Mark Fuccio, previously of Drobo, if any of you remember that storage company. We used to work with Mark, and he gave us a few to give away. It was really cool. Good to see you, Mark. Can you explain what human leukocyte antigen does? I read people that have it are asymptomatic. So we all have human leukocyte antigens, right? Another name for it is the major histocompatibility molecule, which are present, there's types 1 and 2, and they're present on either all cells or simply certain types of antigen-presenting cells like macrophages and dendritic cells. So what they do, the cell is continuously chopping up proteins inside and shipping them to the cell surface, and they sit in these MHC molecules, these leukocyte antigens. And then T cells and other cells can come and look at them and ask if they're foreign or not. And if they're foreign, then they kill the cell, or the T cells do other things. And so the people who are asymptomatic, a good fraction of them have a specific kind of MHC or HLA, because HLA molecules in the human population are highly variable. And so, your HLA is different from mine. And there are a number of different types, just like there are a lot of different T cell receptors. There are a lot of, and antibodies, there are a lot of different HLAs. And so people who are asymptomatic have a certain kind of HLA, plus they were also infected with a common cold coronavirus years ago and have T cells that recognize a certain peptide from the common cold coronas that is the same as in SARS-CoV-2, and that peptide is presented on this special HLA molecule. Okay, so only a certain number of people have this special HLA, and that gives them the ability to present this peptide from common cold coronas that cross-reacts with SARS-CoV-2, and therefore the T cells can eliminate the infected cells, and they have an asymptomatic infection, or at least that those two properties, that particular T cell epitope and those particular HLAs, they correlate with being asymptomatic. And presumably the mechanism is that you can readily eliminate SARS-CoV-2 virus-infected cells. Okay, I hope I did that well. All right, this is the thing I've been looking for, cinnamon gum. Have you ever had a rusty train wheel in your mouth, Doreen? Like, how would you know? It's like in the first Harry Potter movie, do you remember they're eating jelly beans and Dumbledore eats one. He says, earwax. It's like, how would you know it's earwax? You've been eating earwax lately? So, Doreen, you've been eating rusty train wheels? Anyway, cinnamon gum. Okay, people always ask me what it is. It's cinnamon gum. Hello, Rima. Saturday you will try to be on the stream. Yeah, so it'll be, I don't know, sometime in the afternoon in Salt Lake City. Simply because I'm there. So hopefully we get 30, 40 people. Usually it would be nice to see you, but I understand about doing something else. Okay, here in Italy, lots of farmers are freaking out about swine fever, rapidly spreading among pigs and boars. So swine, is it African? Swine, because I think that's the one that's devastated pigs and others. That's like a pox-like virus. It's a large double-stranded DNA virus. So let's just look for swine fever in Italy and see what we pick up. First farm in the north tests positive for African swine fever. Yes, August 17th, 2023. And that's a problem because the virus, this is in Lombardy. And it's a problem because it's been spreading in other countries and it's devastating. There's very little spillover risk as far as we know, right? Because we've never seen a spillover risk. But it's a problem for agriculture, right? So that's why we don't like that to happen. Okay, so Bully said, I don't think I will ever take Paxlovid. I'm not 18, no comorbids. Yeah, you don't need to probably. So that's fine. I think that's great. All right, and I'd have to be really sick to even get a PCR COVID test. And then if you're really sick, it might be too late to even take Paxlovid. But I think you're fine. That's good logic. Did I see The Last Supper? L'ultima cena. No, I didn't. I had a graduate student who always used to say that in the lab. I don't know why. It's kind of a joke. If you didn't get the experiment to work, it's going to be your l'ultima cena. His name was Nicola also, actually, my first graduate student. Anyway, I didn't get to see it. I did see it years ago when I was a kid with my family. And I did want to go back, but, I just didn't have time. I didn't stay long enough, unfortunately. I had planned to go to the Duomo, right? So I got in. I was going to check my bag and then go to the Duomo, but I got to the hotel and they said, your room is ready. So I went and took a shower, took a nap for an hour, and then went to the meeting, which was at 3 p.m. anyway, so it's not like I had a lot of time. So anyway, I didn't see it. Will Amy still send in the Amy papers? Yeah, she's sending me them regularly. I haven't posted in about a week. But yes, she still does it, so you can check them out. Yep. Smiles with your eyes. Smiles with your eyes are so nice, aren't they? And I think when you cover the mouth, I'm smiling with my eyes. Yeah, that's cool. I like that. Smiling with your eyes. Very, very attractive. Yep. Yep. I'm 42 in New York and got prescribed Paxlovid this morning. Is that weird? Or did they ask you if you wanted it, or did they say you need this? I don't, it's not weird. No, I know a lot of people who got it at younger age. Yeah. Even younger. I mean, I know very, very young people who got Paxlovid. I mean, I was 69 when I got COVID, and I wanted to see what would happen. And Dana said, no, you shouldn't. Even though I have zero comorbs, healthy, very few colorless hairs. And he said, you should take it. Don't take a risk. I really wanted to see what it would be like, but you don't do experiments on yourself. It's silly. Yeah. It's not really smart. I don't know. I'm not sure the smile would be as good without the mask, because the eyes smiling are really cool on their own. I mean, it could be a closed, a very tight mouth. It could be big, and who knows. But I like just seeing the eyes smile. It's very cool. Is it me that finds it ironic that people who study viruses happen to be the most vibrant and healthy group of people I have ever seen in photos? Oh, based on those two young ladies smiling? That's because they just met me, dude. Whatever Wondering World is. Can I make that joke? No, they were very happy to meet me. And then there was a lady standing by after they left, and she came up and she said, I didn't know you were famous. No, it's just viruses. But I'm not going to say that everybody is happy and smiling. If some people, when you go up to them, they smile, they're happy to meet you. But I don't think it's restricted to virologists. On the other hand, they are happy studying viruses. It's really cool. Anyway, the toilet surveying is interesting, because I said to her, do you have to get permission from China? And she said, no, it lands on Belgian soil. We can do what we want with their poop. She actually used the word poop in her talk. I thought that was funny. I said, it's not really poop. It's a mixture of stuff, right? It's toilet paper, it's urine, it's poop, and the chemical that they put in, that blue stuff, right? It's a mixture of stuff. Well, here we have good evening. It's 64F, waning crescent moon phase. Subvariants EG5 and FL1.5.1 are currently dominating in the U.S. Thank you, Patricia, for that report. Yes, Jolene adds as phage specialist. I really like having a phage person. Yep. Are there any illnesses out there that can cause you to lose your appetite? Now, I'm not going to talk about non-viral illnesses, because that would be out of my lane. But many people, when they get a fever, don't have an appetite. So I think if the viral infection is inducing a fever, which many do, you could lose your appetite. Certainly gastrointestinal viruses like neuroviruses and rotaviruses, which make you vomit and have diarrhea, you probably don't have much of an appetite when that happens, right? I would think. So many of them do, yeah. You can't diagnose them, certainly on that. Okay, any thoughts on using poliovirus for solid tumor treatment? So there's a phase 2 clinical trial for glioblastoma. So I'm not sure I have so much enthusiasm for this. Now, the problem, of course, is that we don't have any treatment for glioblastoma, so any improvement is better than nothing. And there was a marginal improvement on survival of patients that were treated already with poliovirus injected into the tumor. I don't understand how, well, now that I think poliovirus will not be eradicated, then I guess it's okay to use poliovirus for these treatments. But I don't see that it's going to result in a cure based on the original studies. Yeah. So I'm not really big on that at all. I am not. My brother used to work at the airport. They called that job honey dippers. The ones that drain the waste out. Oh my gosh. Oh my gosh, that's something. Brandon, how might advancements in metagenomics and sequencing technologies help us uncover this hidden viral diversity of viral dark matter, and what could be the consequences of doing so? Well, we have a really good metagenomics pipeline already. So, I mean, we have FAST. We have fast sequencing, right? We have great bioinformatics tools that we can do to analyze it. I mean, you could improve the error rate and so forth. You could improve the read length. However, how is this going to help us uncover? You get sequences, and no matter what their quality is, you don't know what they are, right? And so I think that will not help you identify the dark matter. The way you can do that is, well, for example, for the crassphages, they found they pieced them together so that they get long open reading frames, and then started probing different kinds of specimens from people, and found that most of us have this phage in our intestines, and so they could identify it as that. So I think that's the key, and I don't think that any new technology is going to help that, right? It's just a matter of going through the data and deciding what you're going to follow up with. So this is not something I think about all the time, but I do know what's out there and how it's used. So I think it would be good to, it would be great to identify, but I'm not sure that it helps. That's my bottom line there. Okay, well, could you talk about the fad capping of hepatitis C virus? So Hep C virus has recently been found, as of July, to be capped with flavine adenine dinucleotide, FAD. So as you know, many RNA molecules that are translated have a 5' cap, which it's typically G, PPPG. And there's some methylations on them. And the Hep C genome is not known to be capped, but they found in this paper, this was published in Nature, that it's capped with a non-canonical initiating nucleotide. It's capped by the RNA polymerase of the virus. So you have a flavine adenine dinucleotide on the 5' About 75% of the RNAs have that cap, which doesn't stabilize the RNA, but protects it from being recognized by innate immune systems. So what's your question? Is new research that hypothesizes how it establishes chronic infections. Yeah, I mean, they say it's a new strategy, could affect persistence of infection, but because the idea would be that it's protecting the RNA from being recognized by innate immunity, right? Because the recognition of RNA is dependent on the 5' end in great part. So it could, but I think they need to do studies to show that. And as many would argue, those studies are going to be done in cells and culture, so who knows what the relevance might be. So I think, Tom, it's just a speculation because of it's at the 5' end and could mask the RNA, but I agree that we have to see. I don't know if it's right or wrong at this point. Wondering world, I think the world is so connected with flights that maybe we need to start wondering if one country is endemic, then the world is one vaccination scare from a pandemic. You mean if one country is endemic for some virus that and nobody else is, that it could easily spread? Sure, but what's the likelihood that there's no immunity elsewhere, right? I think it's pretty low. I think the scare is from viruses that aren't in us at the moment, right, for which there's no immunity. I think that's really the issue. This is good. If you want to get rid of Hep B, you can use a HEPA filter. Well, that would get rid of Hep A, right? It's Puscast, Billy. Infectious disease Puscast. Yep. Which Patricia said, yeah. Hepatitis, and Tom is hot tonight. Hepatitis in Dutch meat follows the fecal oil route, giving the name to the Low Countries. Very good. Is hepatitis E not inactivated by cooking? It is, but many people like their meat, raw, not raw, but medium, and so that leaves some infectivity there, yeah. How many respiratory infections is a lot? Well, that's a good question. I would like to get the figure, and I'm going to do that. I haven't had a chance. But from the graph he showed, we're not talking about 50 or 100. We're talking about less than 20 versus 2 or 3. But that's a good question. I'm going to get the paper, and maybe we'll do a snippet on TWIV. I think it's worthwhile. Yep. So, yes, this is a good, interesting idea that then if you are going to live less, if you have a lot of respiratory infections, then maybe you should wear a mask in the respiratory virus season, and you could live longer. Oh, what's an interesting question? Okay. Yeah, that's another one. What constitutes a lot? I guess I should have not said a lot. My son contracted Entamoeba histolytica, and we never left the country. Any idea? Contaminated feces. Diarrheal stool. Frequently covered from fresh water contaminated with human feces. Majority cases occur in developing countries. So, there are certain risk factors, Noir. You might want to look it up, but it's something you would acquire from fecal contamination like poliovirus or norovirus or rotavirus. I wonder if there's a difference between upper and lower respiratory tract infections in that study. It was lower tract infections, if I remember, John. Yeah, I didn't get the reference. It's published on the 7th of March, 2023, so I'm going to use some fancy keywording here to try and find it, because he changed his slides too quickly for me to write it down. Right? Did you ever have that problem? Recent Nature paper proposes that persistence in various tissues is a main cause of post-acute sequelae of COVID. I don't know if it's right. Not clear to me that you need to have persistence to have. How would you show that, right? So if you said everybody with long COVID has persisting antigen, well, nobody's done that study. So still, how do you know it's a cause and not something else? I mean, you could speculate, that's fine, but I don't think it persists in everyone who has long COVID. I think there's some kind of association, but, we're going to need some models of this, and we just don't have them to figure that out. And, whether it's just antigen or infectious virus, you don't know. So I'm just a little skeptical about that. Just finding antigen in the tissues isn't really enough. Patricia's thinking about getting Shingrix and RSV vaccines. So I got both Shingrix. I think it's a good idea. And RSV I will get, yeah. Because RSV can affect many people and can be a serious infection. And the more serious respiratory infections you get, the less years you're going to live. So it seems to me that's a good reason, right? I've never watched a football game in my life. Good for you. Which kind? American football or soccer? Should my 95-year-old mom get the RSV vaccine? Well, I don't think we should give medical advice, but you should ask her doctor. But RSV can be severe in elderly people. Your mom's not a youngster, and it could be recommended. That would be, she is the target population. Whether or not she has other issues that would prevent it, I don't know. So you have to check on that. But I think it's a good idea because the very young babies and the very old people are the ones who are at risk for the most severe disease. And but ask your doc. I guess what we can say is if you want to live a longer life, better ventilation is important, but don't try to achieve the same by clearing your airways with your digits. Yeah, don't put don't put your fingers in your nose. You get more respiratory infections, and that's a problem. Paul, often you discuss decline in childhood vaccinations. Repercussions are likely seeing a rise in measles. If you're an adult who had measles as a kid, do you have immunity? Mm-hmm. You do. I had measles as a kid, and I never got the measles vaccine. And now that doesn't mean anything what I did, of course, but measles is one of those viruses that we think confers very long immunity. So I think, yes, you're protected. This is very interesting. Yeah, so cases of measles are on the rise because there's repercussion. People are not wanting to be vaccinated because they got mad that they had to be vaccinated against COVID, right? So now they're pissed off. Go figure. And now we had almost gotten rid of SSPE in the U.S., right? You guys know about SSPE? Long-term sequelae of measles virus infection. 1 in 10,000 kids, 10 to 20 years after recovery from measles, they start to degenerate neurologically, and eventually they're dead. 100% fatality. 1 in 10,000 cases of measles. We had almost eliminated that in the U.S. by vaccination. Now it's going to go back up again because kids are getting measles, and 1 in 10,000 of them are going to get SSPE. Why is this smart, folks? Because you got to choose? And your kid didn't even have to choose, didn't have any role in the decision, right? And you're going to be responsible for saying no because you think that these vaccines are causing autism, which they don't. You're not going to vaccinate your kids. It's ridiculous. It's totally ridiculous. But it's your life. It's your life. I read about a new mosquito species that likes indoors. How about bird? Well, influenza viruses don't get vectored. At least human influenza virus, avian, don't get vectored by mosquitoes. There's no evidence that mosquitoes are a vector. So this is an interesting thing. So I said they don't get vectored, but can we say that they never get vectored? No, you can never say they never get vectored, but it's the limit of science, right? You can never say never. But we've never seen any examples of vectoring of these influenza viruses by mosquitoes. So I'm not worried about that. And, I asked Ron Fouché, can people have bird feeders? He said he thinks it's fine. He's not worried. In fact, he's not even worried about avian influenza as far as humans go. He thinks wild, non-human animals, other mammals, are at risk because they're big die-offs of sea lions, even marine mammals. So they're worried about that. But he's not worried about people. And he's worked on H5 for his whole life. So I'm not worried about the mosquitoes either. Well, is this addressed to me? I don't know what vaccine you mean, Wanda. No. I'm not getting any vaccine. Do I need a vaccine to China? Let's see. I don't think so. No, I don't think so. I got a yellow fever vaccine before I went to Brazil a couple of years ago. And that's it. But I don't think so, no. Any immediate RSV vaccine side effects? Yeah, there are side effects. And, your usual pain at the injection site, fever, nausea, etc. One of the two vaccines had more side effects than the other. And it may be related to how much the adjuvant or the dose, I don't remember what it is. Pain, redness, swelling, fatigue, fever, headache, nausea, diarrhea, muscle, joint pain, usually mild. One of the two had more of that than the other. So the GlaxoSmithKline RSV vaccine is adjuvanted, and the Pfizer is not. And so I think the adjuvant vaccine was associated with the slightly more severe side effects of those that I just mentioned to you. So if you would like, try and get the other one. What do we have here? 204 people. Thank you for coming. 140 thumbs up. Please hit the thumbs up button. That would be great. Why? Because we could attract more people to this. My goal is to teach more people. I like teaching. I like when people learn. So okay. What is the difference between having live virus and just the sequence? It was unclear on this when hearing about the Wuhan Institute of Virology not having infectious RATG13, even though it was too different from SARS-CoV-2 anyway. Well, infectious virus. Here, I have a tube here, a 50 milliliter conical tube, right? And yeah, I brought this from my lab because I like these. I think these are very cool. but we can put virus. This is clean. There's nothing in it, but we can put virus in it. So let's say I give you here. I'm going to take my Twiv mug and I'm going to pour some water in just to illustrate. So here we have a tube. I'm going to hand you. This is infectious poliovirus. It's not. I don't have any infectious polio. It's just an illustration. Influenza virus. Here, take this and infect some cells with it. So in this are virus particles that can bind to cells and initiate an infection. That's infectious virus. Now, having the genome sequence is a series of 4 bases, right? C, G, U, and A on a piece of paper that specifies the genome of this virus. What are you going to do with that? Well, Racaniello showed in 1981 that you could make a DNA copy of it and put it in cells and it would initiate infection. So you could do that. But for coronaviruses, that doesn't work. If you just make the DNA and put it in cells, it won't give you. You have to do some very special things. And it's hard to get the big DNA anyway. So the genome can be infectious. But it's not as efficient as having virus. But in the case of RATG13, that genome wasn't infectious and they had no infectious virus. So they did try and put it on cells and they never got virus out. I don't think they had the right cells for that particular virus. So that is the difference. The virus particle, right? It's a membrane encased particle with the genome in it. It's very infectious, much more so than the naked RNA. The naked RNA can also be infectious, but the main point about not having infectious virus is that you can't grow the virus. You can't propagate it. So you can't do any experiments of infecting cells and infecting animals or whatever it is that you want to do. So that's the main problem. But that turns out that RATG13 is just not, they don't have the right cells for it most likely, or maybe the genome sequence is wrong. Tom says, with respect to electron microscopy, also there would be controls, this is correct, of uninfected cells, organisms, corresponding biochemistry, molecular biology evidence, antibody binding, nucleic acid sequences. Right, there would be other things as well. And you could put them all together, right. You could take those things that look like viruses, purify them and see if there are proteins in them, if there are nucleic acids and so forth. Yeah. Are the colored 3D pictures graphic representations of black and white microscopy images? It depends. Sometimes they can be. Sometimes they're computer generated, right? So, some of the Mimi virus pictures I showed you, I think they're all either based on an EM or drawn, because we don't have a structure for that fiber. So those have to be, drawn by an artist. But the icosahedral capsid, I think I have one here for polio. Okay, so here's a 3D printed polio. It happens to be a Christmas tree ornament that somebody gave me with no less Twivs stuck on it. This is 3D printed. So this is based on the actual structure of the virus. You get the coordinates and you can make a 3D representation of them. And sometimes the artists draw them by hand. So David Goodsell famously draws viruses by hand. So it depends. It can be both. Yep. COVID seasonality, any large multiple gathering events were super spread. Yeah, but, for flu in the summer, that happens, but you have large gathering events in the summer. People have weddings and parties, and you don't see outbreaks of influenza in the summer in temperate climates. There's always a little bit of flu, but they're not outbreaks. So that's why we say the seasonality is in the winter. So that's not the only explanation there. You had RSV last year. Should you get the vaccine? I think Daniel has been asked this, and he said you are good to go if you've had it. And I know some people who have had RSV infection and are not getting vaccinated until next season. So, yeah, that's what Daniel says. I listened to your interview with the two scientists, virologists. I get confused with all the different vaccines. Oh, that must be Kostya Chumakov and Raul Andino. Yeah, I'm sorry. The sound wasn't so great on their end either, right? I mean, I edited the audio on that, and yeah, they had crappy microphones. They're talking into their laptop. I don't know what to do about that. Anyway, yeah, it was a confusing discussion. It was kind of a high-level discussion. The bottom line is we're not going to eradicate poliovirus. We're going to have to keep immunizing forever to control poliomyelitis. And that this vaccine will be used in some countries, and most countries will not use it, which is one of my problems because it causes polio. And I don't think any country should use a vaccine that causes polio. But, WHO tells most of Africa, here, use this vaccine, they use it, and then kids get polio. But we'd never use it in the U.S., never going to be used in Europe, and that's what they said in that program. I no longer think it's acceptable to paralyze kids with a polio vaccine. Fix it. Make a better one. That's what they tried to do, but it's not fixed. It still causes polio, even though it's reduced compared to the original oral polio vaccine. The thing is, it's easy to give, and that's why the default is to give it, and you just drink it. But I think we should make a better one, but we're going to stop polio research, so we can't, and that's the silly thing that's going on there. All for cockta, right? Did I use that well? Like hurricanes name new epidemics after anti-vaxxers and science deniers. That'd be good. Part 2. I only remember the Sugar Cube 60s. Do we need a new one? True, it takes 10 to 15 years after infection to paralyze. No. The vaccine-induced paralysis happens within weeks after immunization. In fact, 7 to 14 days. So, and it doesn't spread by houseflies. No, that was tested actually, and we never saw it spread by houseflies. Nope. What's the Mimi virus receptor binding domains? Do the hairs aid in antibody evasion? I don't think the hairs have anything to do with receptor binding. And the receptor is binding to the capsid. I'm pretty sure the receptor is not known. It may not actually have a specific receptor because the host, which may be amoeba, are just taking things up by phagocytosis, right? So I don't think that's a worry. That's something. There's no RBD. Do the hairs aid in antibody evasion? Yeah, I think amoeba don't make antibodies, right? So probably not. And I'm not sure it infects hosts that make antibodies. I'm not sure. There's some mixed data on that, but I'm not convinced that it's doing it. So there you go. Could be a question for Dr. Griffin. If you have hypertension that's well controlled with meds, you consider that comorbidity. I don't know. A COVID comorbidity. Well, I'm recording the clinical update right after this. Hypertension has been identified as the most prevalent cardiovascular comorbidity in patients infected with SARS-CoV-2. that demonstrably increases the risk of hospitalization and death. So there you go. The answer is yes. Now, you're saying it is controlled, right? That's a good question. See, that's the part I don't know. Should all patients with hypertension be worried about developing severe COVID? You know, this study looked at hypertensive patients over 60 years old with a BMI over 25 kilograms per square meter, etc. They all had poor outcomes. And so I think the answer is that yes, if you are hypertensive, yes, you're at severe risk no matter what. But could ask Daniel. Yeah. Are these names official? I think the only official names are the dot names, because those are put out by the, next strain and covariants, etc. But, WHO seems to be using the friendlier ones, which I don't know why you have to confuse things halfway through. TWIV 1028 showed a common allele of HLA mediates asymptomatic SARS-CoV-2, right? Cross-reactive T cells, exactly. Does this rule out potential asymptomatic infection via innate immunity? No, it's not everyone who has an asymptomatic. I think it was only 20% of those people who have asymptomatic infection. So there are other mechanisms at play, for sure, and that could be one of it. Since vaccines don't prevent infections but prevent disease, how does the measles vaccine eradicate measles where it's used? That's a good question. I don't know the answer, and that nobody has studied it enough to know. I think it reduces transmission most likely, but you have to keep up immunization. Otherwise, you do have outbreaks. So there is some circulation, obviously, right? Love the alpha-gal paper on TWIM. I've been fascinated with the rise in alpha-gal syndrome, which is a tick-borne disease. CDC has a publication on it. Yeah, it's a very cool story there. Go check it out. Donald Rumsfeld, thank you very much. Bowden has listened to TWIV since 2014. Following in your footsteps, I'm working on an online bio course for students. Cool. Can I consult with you? Absolutely. I'm happy to help. Vincent at microbe.tv. Let's see, we're getting to the end here. But if you aren't very contagious until you have disease, then preventing disease does drive infection out. So that's what happened with SARS-1, that you weren't contagious until the peak of disease, and so that was easy to put people in a hospital, do infection control, and so forth. What about BA.2.86? Is there a huge difference? There is a huge difference in spike. There doesn't seem to be any evidence that it is. What are you asking? Is it more virulent? There's no evidence that it's more virulent, and I don't even see any evidence for any of the other variants being more or less. You can find studies that say the opposite for the same one. So these observational studies are just not telling us anything. And it doesn't seem to be more immune evasive. So I think the current feeling is that it is going to be addressed by antibodies induced by the XBB.1.5, right? My daughter called the test. She had nose running. Her COVID test was negative. Yes, wait 2 days and test again. Absolutely. When you get a negative antigen, test again. That was the outcome of the paper that Daniel discussed some time ago. How do other cold viruses get into cells? Oh, the many different viruses can code. Yeah, I just did my nose. You see that? So I have to wash my hands before shaking hands. We have coronaviruses, we have paramyxoviruses, we have enteroviruses, we have rhinoviruses, adenoviruses. They're all different viruses. They all bind different receptors. They get in cells all differently. Yeah. Yep. On the last TWIV, you talked about the stability of polio vaccines. Does that refer to the stability of the structure or the chance of a mutant appearing? The latter, the chance of a mutant appearing in the stool. Absolutely. Totally. Okay, let's see here. We have 15 minutes. What do we have here? Why do you think people are dissing Fauci? I'm sorry, you have to. Let's look it up. What he said, because I'm not aware of it. You have to tell me what statement you're asking about, Brace. I don't know. When will next generation sequencing be available for diagnosing chronic infections rather than just urgent ones? We've done a couple. Well, for bacteria, it's being worked on, right? So laboratories currently diagnose bacterial infections in various ways. Some are culture dependent, mass spec. They're trying out next generation for that. And the thing about a chronic infection is that, well, you have plenty of time, so it's not hard to diagnose, right? So I'm not sure, it's always a cost trade-off. And so that's going to be the issue, whether it can be cheap enough to use NGS. I don't know. Any info regarding new antivirals for COVID? Yeah, I did see one at this meeting. Let's see what it was. If I'm going to be able to find this again. Nope, that's rituximab. That's different. Oh, that's CMV drug. Sorry. I heard other drugs, not COVID. I didn't hear anything about COVID antivirals. Nope. Do older adults who are vaccinated for measles still have a risk? No, I as I said earlier, immunity is lifelong, and you don't need to get a booster if you know that you were either vaccinated or had the infection, as I did as a child. What unanswered questions about Yara virus do you find most intriguing? Grant McFadden used to talk about that. It's another amoeba virus. I want to know what it's. Well, there are a lot of genes in it that don't have any known function. I'd like to know what they are. No recognizable genes. I think that's the biggest question. Genes never seen before. That's dark matter. I'd like to know where it came from or what it really infects. Dr. Griffin has talked about RSV vaccine for children. Is there an approved RSV shot for adults? Just elders. Yeah, just the elder. Not you, Mark. You're young. Let's see what the age cutoff, right? 60 years and older, recommended. Yep. 60 years and older may receive vaccination. So that's not elder, right? It's elder is 75 and up. So you could, if you're 60, you could get it. I can get it. I will. Yeah, there is no animal model for long COVID. Some people have had some suggestions of things, but nope, not happening. People also not wanting to be vaccinated because of the endless drumbeat of anti-vaccine cracks. Yeah, I think, but Paul has said that the COVID mandate has pissed a lot of people off. And the many countries in Europe did not have a mandate for COVID vaccines, and yet many people got vaccinated. So I always wonder now what would have happened if we had done the same. If I remember right, a virus has to survive to transmit by them. It has to actually multiply. Most vector-borne viral infections, the virus has to reproduce in the vector. There's some exceptions where you have mechanical transmission, right, on the mouth parts, but most have to reproduce. The mosquito takes a blood meal, it goes to the gut, and it reproduces in cells, gets into the circulation, then goes to the salivary glands where it goes back out again. So that has to reproduce. Yeah. I had chickenpox also as a kid, and I have a big scar still on my chest from it. Because I remember it was a huge blister on my chest, and it went away eventually, and there was always a scar, and I can still see it. It's so weird. Very interesting. A mini survey. What was the worst infectious disease you'll ever got? I never had a vaccine that gave me bad side effects. And the worst infectious disease, I would say, I had rotavirus 15 years ago. It was horrible. Vomiting constantly, which is, and the dry heaves. Oh my God, that's so awful. When you feel like crap, right? You have a pain in your gut and you have to throw up and nothing comes out. That was bad. I never had any bad flu. It's interesting. So a lot of people are recounting their experiences. What's the status of an Ebola? There are 2 licensed Ebola vaccines. So that's done, and that's for the predominant Zaire strain, and then they're making Sudan strains now as well. So yes, and when there are outbreaks in Africa, they can go in and vaccinate with that. Good question, Bully. That was good. It got a lot of people talking. I like hearing the joint efforts on polio during the Cold War. Science brought people together for common good several times, and that was one of them. Yeah, that's absolutely right. Can I bring Rudy Yanish on? I really dislike what he did with the integration of mRNA vaccine material into the genome. I really think that was just bad science. So I don't want to do that. And maybe he has certainly done other things, but they don't have to do with viruses. I wonder if class of drugs with which the HTN is controlled would make a difference. I thought ACE inhibitors were looked at and they had no effect. Right? That's my recollection. Am I going to take the booster? No. I've had 3 ancestral vaccines, and I've had an infection last December, and even though it's not Okay, Daniel is texting me. He wants to start recording. Okay, Daniel, I'll wrap this up. I feel that I'm sufficiently protected, and if I get infected again, I will take Paxlovid. All right, I'm going to wrap this up 5 minutes early because Daniel wants to, he's ready when I am to do his clinical update because tomorrow I'm in Salt Lake City. All right, folks, thank you for coming tonight. I really liked being here with you again. I missed you all. I want to thank the moderators, Vanity Nutrition, Les, Barb Mack, Tom, Frank was here. Thank you all for being here and moderating. I know it's a long show and it's not easy to do. I want to thank everyone from Coving. 189 likes out of 206. Not bad. Thank you for that. We will be back next week, and I will have a guest. So make sure you mark your calendars. 8 p.m. next Wednesday. That is September 13th. And thanks, everyone, for coming. Really enjoyed it tonight. Great questions. If I didn't get to yours, bring it back next week. Meanwhile, stay safe, folks. Thanks for coming. Good night.